Great. Good morning or good afternoon, depending on where you are. I'm Andrew Van Camp, and today I'll be talking about a preliminary annotation of the MHC of a squamate lizard, Anolis carolinensis. The MHC encodes genes important for immune function, like antigen recognition and signaling. And the MHC is of interest to evolutionary biologists because it has the greatest nucleotide variation in the human genome and in other genomes. This figure here is just showing nucleotide diversity in one locus in red from a random spot in the human genome and the MHC on the right, and we see much greater variation. One of the results of that variation is that it's more difficult to resemble MHC than other regions, as Scott was alluding to, which will be a theme in my talk as well. So the MHC has been pretty extensively studied in mammals and birds, but not in squamates, lizards and snakes. If we take a look at the number of species in each clade versus Google Scholar hits for birds and MHC, just as a rough estimate, we see birds 10,000 species, 30,000 hits, mammals 6,500 species, 90,000 hits, but squamates 7,500 species, only 161 hits. So the MHC has been underexplored so far in squamates. So we wanted to try to take a preliminary look at the MHC in a squamate. And they should really be a pretty interesting clade to examine the MHC in. They're a clade of evolutionary transitions and convergences. For example, all squamates are ectotherms as opposed to mammals and birds, which are endotherms, which could have interesting consequences for the MHC. Within squamates, we see a variety of different sex chromosomes, which again could have something to do with immunity. And also within squamates, you see a mix of oviparity and viviparity. Again, there could be some implication for immunity there. So we chose to look at Anolis carolinensis for this first look at the squamate MHC because it's a model squamate. It's been studied for decades by ecologists and evolutionary biologists, and it has a high quality genome assembly, the first actually in a squamate, and it has a rich history in comparative genomics databases like NCBI and Ensemble. So we set out to just answer a few basic questions about the MHC in the green anole. First, where is it? And we saw the chicken MHC is on chromosome 16, and the paper was saying that chicken chromosome 16 displayed synteny with anole chromosome 2. So we hypothesized that the anole MHC would be on chromosome 2. And we also wondered if we could use a lot of the information that people have found in mammals and birds and apply that to the anole MHC. So looking what genes from the human and chicken MHC are in the anole MHC. And in terms of size, the human MHC has more than 200 genes and the chicken MHC has 19, which is minimal essential. So we hypothesized that squamates would probably be more similar in size to humans than to chickens. So to answer that first question, where is the anole MHC located? We detected homology with BLAST. We made a list of MHC genes from a couple papers and pulled down the squamate proteins corresponding to the listed genes from NCBI for about 2,400 records total. And we aligned the protein sequences to the Anolis genome with the tBLAST10 algorithm. And when we binned those hits by chromosome, we got this figure showing that the majority of our hits were on chromosome 2, which shows that the anole MHC appears to be on chromosome 2, but there were also a good number of hits on unplaced scaffolds, which we think might actually be potentially part of chromosome 2, but just a poor assembly quality at this point. So to refine this analysis a little bit more, we calculated and plotted how many hits we had per megabase pair sliding window. And when we separated into class 1, 2, and 3, we see that they're actually at the very end of the scaffold, which could indicate that yeah, it's just an assembly issue. The actual chromosome actually goes on for longer, but we've just only been able to assemble it to this point with the genomes that we have at right now. So to take a look at that second question, what can we learn from the information that we have from mammals and birds? We inferred orthology with Ensemble. We queried Ensemble for our list of genes and recorded the orthology data that they had already compiled for them and created this synteny plot. So just to orient you, we have the human MHC on the top and the anole on the bottom left, the chicken on the bottom right. The green rectangles are coding regions, and the orange lines connect genes that Ensemble dubbed as having a one-to-one relationship, and the blue lines are those that Ensemble said had a one-to-many or many-to-many relationship. So you see a good amount of one-to-one orthologs, but you also do see a lot of one-to-many or many-to-many relationships. And what that looks like, if we just zoom in on one example, is for this gene called MIC-A, anole has 2 copies of it, human has 2 copies, and Ensemble is unable to resolve which paralog corresponds to which paralog. So to try to refine this analysis to maybe see if we could resolve that, we conducted a reciprocal best blast in which we pulled down the human proteins corresponding to our list of genes from NCBI. We aligned the protein sequences to the Anolis genome with BLAST. We aligned the top hit back to the human protein sequences with BLAST. And if we ended up at the original gene, we dubbed it a reciprocal best blast hit and created a flowchart with our results. So we started with 143 human MHC genes that Ensemble, 62 of them had been characterized as one-to-one, 23 as one-to-many or many-to-many, and 58 as no homology. And out of those 23, there were 4 single protein genes and 19 multi-protein genes. Of the 19 multi-protein genes, we found 6 additional reverse best blast protein matches. And of the 58 with no homology, we found 1 additional reverse best blast protein match. So our analysis was able to find orthology for 7 more genes. So going back to this synteny plot, one other feature of it that we noticed is that the anole MHC is much more spread out than chicken or human. In this figure, the line is just longer. And that's even potentially without having class 2 there. Recall that the assembly ends here at the end of class 3, but even looking at the pattern in human with class 1, class 3, class 2, it seems like perhaps class 2 fits in there on those unplaced scaffolds. But the anole MHC is longer, and we were wondering why. We thought maybe it had to do with structural features of the genome, like so we asked, how do intergenic lengths compare between anole, human, and chicken, and between MHC regions in the entire genome? And how do intron lengths compare between anole, human, and chicken, and between the MHC regions in the entire genome? So we found that it was that the anole MHC was actually more spread out due to both intron and intergenic length. So this violin and box plot here is for intergenic length. To orient you, we have the anole whole genome, anole MHC, chicken whole genome, chicken MHC, whole human genome, and human MHC here. And the p-values are shown for only those pairwise relationships which were non-significant. So if you don't see a bar, the comparison was significant. So what we see in chicken and human is a decrease in intergenic length between the whole genome and the MHC. And we see the same thing in anole, but it doesn't decrease by as much. If you look at the means, it's about a decrease of 0.4 in the log scale on these and 0.2 on these. And we see a similar pattern in the intron lengths. This is basically the same figure, but with the intron lengths, we have our anole, chicken, and human whole genome versus MHC. The drop-offs from chicken genome to chicken MHC and human genome to MHC are greater than that from anole genome to anole MHC. So this implies that there might be some structural differences to the anole MHC that make it fundamentally different from chicken or human in some as of yet unknown way, or it could just be, again, issues with our assembly that new genomes might resolve. So to conclude, we found that the anole class 1 and 3 MHC is on chromosome 2. We were unable to locate the class 2 MHC except on some of those unplaced scaffolds, which we think might be a part of chromosome 2. But I will have to wait for some long read genomes to find out. We found orthology for 69 out of the 143 human MHC genes we tried. So future work will involve continuing to clarify orthologs and investigating genes unique to the anole, and of course, improving our gene annotations and assembly quality in MHC regions. And so the whole idea of the line of this work is to eventually investigate MHC differences between phenotypes that can only be studied in squamates, like that of endotherms versus ectotherms between mammals and birds and squamates, and oviparity to viparity within squamates. With that, I'd like to thank Scott Edwards and Darren Card for all their help with this work. And some other Scott Gustavo Bravo, Flavio Tamagnoni Garcia, my funding, the Harvard Center for the Environment, and the organizers of the symposium. And I'll be happy to take any questions. Thank you.